Welcome, everyone. This is the Myotonic Dystrophy Foundation's Meet the DM Drug Developer series. Today is Wednesday, June 9th, and we are here to meet ARTHEX Biotech. As a reminder, the MDF mission, Care and a Cure, is to enhance the quality of life of people living with myotonic dystrophy and accelerate research focused on finding treatments and a cure. Our work focuses on support and education, research, and advocacy. A reminder of the support materials and opportunities that are on our website. We have resources such as toolkits and publications, including clinical care recommendations, virtual support group and Facebook chats, our calendar of events and activities, and our digital academy, which includes presentations and videos from past events, as well as today's presentation will be posted either later today or in the next day or two. Okay, just like today, we have our Meet the DM Drug Developer series that usually happen the first Friday of the month at 12 o'clock Pacific. Next month, we will be visited by Harmony Biosciences, and then we will hear in August from Avidity Biosciences. Ask the Expert series is coming up third Friday of the month at 12 o'clock Pacific. This month, we will hear from a speech-language pathologist who will be talking about speech, language, and swallowing with myotonic dystrophy. We just started to confirm our final two presentations for Ask the Expert series. So July, we will be hearing from an expert on the brain or central nervous system, and then we'll be hearing from someone focused on anxiety and myotonic dystrophy in August. A reminder to those who have heard and those who have not yet heard, we have declared International Myotonic Dystrophy Awareness Day for September 15th. You can learn more about the Global Alliance and how you can get involved at myotonic.org/international-DM-day. As mentioned, all of our sessions are recorded. They will be posted to our digital academy at myotonic.org/digital-academy. If you have questions about today's presentation, you can contact MDF at the phone number here or via email at info@myotonic.org. Okay, about today's presenters, we will be hearing from three people from ARTHEX Biotech. Beatriz Llamusi is the ARTHEX CEO and co-founder. She received her master's degree and PhD in biochemistry from the University Miguel Hernández in Spain. During her PhD, she focused in the molecular basis for neuronal regeneration. After that, she began working in the Department of Genetics as a postdoctoral researcher, where she dedicated more than 13 years of research to pathogenesis mechanisms and therapeutic approaches in rare neuromuscular diseases. During this period, she produced 22 scientific articles and 7 related patents. She was the PI in 5 competitive projects and supervised 4 PhD projects. During the initial steps of the project that led to ARTHEX Foundation, she was awarded several valorization grants, including training in business management and development, IPP, financials, and regulatory issues. She co-founded ARTHEX Biotech in December 2019 and has served as the CEO since then. Joining will be Rubén Artero. He is also an advisor and co-founder for ARTHEX Biotech. Dr. Artero received his PhD in biochemistry from the University of Valencia in Spain. During his doctoral degree studies, he discovered the muscleblind gene, the founding member of the MBNL family of proteins, and completed 6 years of postdoctoral training at the Memorial Sloan-Kettering Cancer Center in New York. Dr. Artero currently serves as a full professor of genetics at the University of Valencia and as co-director of the Spanish meeting on oligonucleotide therapeutics, as well as academic editor of the journal Scientific Reports. In the last 4 years alone, he has published or contributed to nearly 20 articles in peer-reviewed journals. As the leader of the Translational Genomics Lab at the University of Valencia, His laboratory is focused on the molecular basis of muscle atrophy and myotonic dystrophy, and in identifying appropriate therapeutic targets and candidate drugs using cell, animal, and bioinformatic approaches. Also joining today is Dr. Nicholas Johnson, who is an ARTHEX advisor. He also serves as an associate professor of neurology and human and molecular genetics and is vice chair of research in neurology at Virginia Commonwealth University, with a focus on inherited neuromuscular disorders. He received his undergraduate degree in molecular and cellular biology and psychology at the University of Arizona. He then obtained his medical degree at the University of Arizona, completed his neurology residency and combined fellowship in neuromuscular medicine and experimental therapeutics at the University of Rochester. Dr. Johnson was also an MDF research fellow. His laboratory at VCU is focused on identifying the pathogenesis of limb-girdle muscular dystrophy, FSHD, and myotonic dystrophy, and identifying appropriate clinical endpoints for these conditions. Dr. Johnson conducts therapeutic trials and many other inherited nerve and muscle disorders, and currently leads the International Myotonic Dystrophy Clinical Research Network Natural History Study. Okay, thank you all for those who registered in advance and submitted questions. Those questions have been forwarded to the team at ARTHEX, who will be talking today. If you have questions during their presentation, please use the chat function in the chat box of GoToWebinar. Select the drop-down menu that says Send Question to Staff and send it. If you do not select Send Question to Staff in that drop-down menu, unfortunately, the question will not reach the presenters. So chat box, drop-down function, send question to staff, and those questions will get to the team. Okay, thank you everyone for joining us today, and welcome ARTHEX Biotech.
Hello, my name is Beatriz Llamusi. I'm very happy to be here with you all today. Here it's 9 in the evening in Spain, but it's a pleasure to share with you our project of ARTHEX Biotech. Okay. As you can, I will give you a little bit of background. Hopefully you can see my screen, full screen. If not, please let me know. Our mission, even before we created ARTHEX, was to develop a treatment against myotonic dystrophy, because we have been working a lot on this disease. And in fact, here I give you a little bit of historical background. The first event here was already in 1995, when Rubén Artero, our co-founder, discovered the gene MBNL. You'll probably already know this gene because it's very involved in the pathogenesis of the disease. When he came back to Spain, he created his laboratory in the University of Valencia, and I joined this laboratory as a postdoc in 2007. Since then, we were working together in different projects, and finally in 2016, we generated results. We discovered that there are microRNAs that are regulating MBNL expression, and they could be potential targets to treat this disease. So we had a patent, then we made a publication in a peer-reviewed journal in 2018. And we also received several valorization and grants that allow us to attract the attention of venture capitals, and then we created the company at the end of 2019. Here I will invite Rubén Artero to stage. Let me see if I can make it, because I would like him to give you more details about all the proof of concept details, all the research that led us to create our company. Okay.
Thank you very much, Tanya and Beatriz, for your nice introductions, and also thank you to all the people who is attending to the webinar. Well, my goal, as Beatriz was saying, is to provide the scientific context around ARTHEX. And to do so, first of all, I would like to introduce you to the team with this picture of almost everyone. Currently, the group consists of 6 PhD students, 3 postdocs, a project manager, and also a technician. Can you still see my screen, right?
Yes.
Okay. I think it's safe to assume that we are all interested in translational research. Well, for translational research, we mean that the process of moving basic scientific findings in the laboratory into potential treatments for the disease. So this process can be split into a number of specific steps. Starting from scratch, we would like to, first of all, understand the pathogenesis of a disease. This is the pathways that go wrong and generate the alterations that give rise to the specific symptoms in a patient. We also need to understand which pathways have to be enhanced or repressed in order to ameliorate the alterations that are typical of the disease. Next, we need to develop molecules that are able to do this molecular job of enhancing or repressing specific pathways. Next, we have to transform those molecules into actual drugs that can be used in patients. And finally, we have to test those molecules in patients in clinical trials. Well, most academic research actually moves between steps 1 and 3, and we ourselves have performed significant contributions in the myotonic dystrophy field, for example, providing the first Drosophila models of myotonic dystrophy type 1 and type 2. We have also proposed the use of miR-7 and the autophagy as a therapeutic target for the disease, and have also discovered a number of candidate compounds, for example, mirtazapine or chloroquine, that for different reasons could not move towards clinical trials. But with ARTHEX, we are hoping to actually transit all the path till the actual testing of drugs in the patients. For the specific technology that ARTHEX is developing, I would like to take you through a tour with all these steps from 1 to 4 that led to the establishment of the company. If we take a flashback of 23 years, as previously mentioned by Tania and Beatriz, during my doctoral thesis, I participated in the identification and characterization of the muscleblind gene in Drosophila myogenesis. And we discovered that this gene had to do with the terminal differentiation of the muscles. Shown here is the pattern of expression of this gene, marked with a specific antibody in Drosophila embryos. Just 2 years later, Maurice Swanson's lab discovered that the proteins, the equivalent proteins in humans, the muscleblind-like, abbreviated MBNL proteins, had a very strong affinity for the CUG repeats that are the genetic cause of the disease. Therefore, this led to a head start in the understanding of the myotonic dystrophy, because it was clear from the very beginning that these proteins had something to do with the pathogenesis. Right now, the molecular cause of myotonic dystrophy is quite clear, starting from CTG expansions in the DMPK gene. When the gene is transcribed into an RNA, the CTG repeats become CUGs, and they get trapped in the cell nucleus, as can be seen here in this with this technique, where the red dots indicate accumulation of this mutant DMPK RNA. The CUG repeats fall into these hairpin-like structures that are the ones that have a strong affinity for the muscleblind proteins. Indeed, there are 2 muscle MBNL proteins important for for the pathogenesis. One is MBNL1 that accounts for approximately 90% of the MBNL protein in the skeletal muscle, and a minor form named MBNL2 that accounts for about 10% of the amount of MBNL in skeletal muscle. So the take-home message here is that a major molecular problem in myotonic dystrophy is that the muscles lack MBNL1. There is a depletion in MBNL1. But the good news is that the genes that encode MBNL1 and also MBNL2 remain perfectly normal in patients. So the typical approach to free MBNL from this aberrant sequestration has been to degrade the MPK transcripts. But our approach has been different. We thought, well, perhaps we cannot, there is no need to degrade the MPK, but we can enhance the expression of MBNL proteins. In order to do that, we turned our attention to small RNA molecules that have a regulatory role called microRNAs. The microRNAs are about 20 nucleotide long, and together with protein complex can pair by complementarity to a target messenger RNA and degrade that target RNA. This happens in about 70% of the human genes and leads to about 20 up to 50% repression in those genes. So in order to discover which microRNAs were might be repressing MBNL1 and 2, we did an experiment in which to human cells we overexpressed a collection of candidate microRNAs, and discovered that when we overexpressed microRNA 23B and 218, there was a strong reduction in the amount of MBNL2 protein, as shown here by these weak bands. And similar experiments with MBNL1 led to the concept that microRNA 23B and 218 were endogenous repressors of MBNL1 and MBNL2. And this actually answers one of the questions that we got about whether the drug we were developing was based on a new approach. And yes, indeed, the approach we are following is different because we are not targeting the MPK, but we are trying to compensate the sequestration by upregulating the expression of MBNL proteins from the endogenous genes. Now that we had repressor microRNAs, the idea was to inhibit those microRNAs, because if we repress a repressor, then we have an activation. In order to do so, we can develop perfectly complementary RNA molecules, as indicated here, with a number of non-natural modifications in order to make the binding of these molecules to the microRNA, the target microRNA, basically irreversible, to make the molecule more stable in body fluids, and also to enhance the uptake by the cells. These molecules are usually referred to as antagonists or antimiRs. So when we treat cells with these molecules, as shown here in different types of cells, in H, I'm showing muscle cells derived from a DM1 patient. As you can see here, in green indicates the amount of MBNL1. You can see here that there is very little expression, and the expression is the signal, it's accumulated in small dots, while in normal cells the expression is higher and takes all the nucleus. In antagomir 23B and 218 treated cells, as you can see, the expression is much bigger than in the M1 cells untreated, and also in this case. Not only the expression of the protein was higher, but also we could demonstrate that the protein was also functional. MBNL proteins control a cell process called alternative splicing, and I'm going to illustrate this process using an example. In English, for example, if you take the letter S and IP, you combinedly, you get the word ship. But if you include an H in between, you get another word and another meaning, ship. Similarly, during the expression of the genes, at the level of the RNA, you can get small pieces of the RNA to be included or excluded, and this leads to different genetic messages. This is different types of proteins. Actually, if we make this example, these two bands here would correspond to RNAs that contain or not small pieces of the genetic messages. And as can be seen here, while in normal cells, you get these two bands. In DM1 cells, the upper band is almost missing. And in antagomir-treated cells, it goes back on in the gels. So this means that not only the protein is, the amount of the protein is higher, but it's also functional. So do we have already a drug? Well, unfortunately, not yet, because some of these modifications have been related to potential liver toxicity and other issues. Therefore, in step 4, as I started the presentation, we have to transform these molecules into actual lead drugs that could be tested in humans. The main issue here is to increase the potency of the molecule and also to reduce the toxicity of the molecule. To do so, we followed two initiatives. The first was the international project TATAMI, that stands for Therapeutic Targeting of MicroRNAs, funded by the La Caixa Banking Foundation, and also established the company ARTHEX Biotech with private investing to move the hit to lead process. In this process, it was needed to generate a lot of chemical diversity. More than 100 variants were generated, and some examples of the different types of changes that can be introduced into these molecules are shown here. There's no need to go into this level of detail, but just to mention that using several in vitro evaluation tests, we have been able to rank these molecules on parameters like toxicity and activity. And the best scoring molecules have been tested or are being tested currently into mouse models. First, the HSA-LR model developed by Charles Thornton, and the DMSXL mouse model developed by Dr. Geneviève Gourdon in France. In the first model, we have tested different doses, also the duration of the procedure, of the treatment, and some of this information has already been published in the literature. And currently, we are validating some of this data in this second in vivo model, and also we're using the DMSXL mouse to test some alterations outside the muscles, and particularly in the central nervous system, for which this mouse is particularly well, it's useful. We are also interested in testing the leading compounds in myoblast coming from a number of patients to test the possibility that the different genetic backgrounds of the patients may have some impact, some influence on in the response to the drug, because it could be that some patients may respond better than others, and this is something that we need to know and understand before going on with the clinical trials. And this also responds to one of the questions we got: what are we working on? We are working on the two mouse models, and also with cells from patients. And to finish the presentation, let me introduce you to the latest and most sophisticated preclinical model we have currently, in which, in collaboration with Dr. Javier Ramon from the Institute of Bioengineering of Catalonia and Barcelona, we have developed in vitro grown human muscle Normally, in conventional cell culture conditions, cells grow in a surface in 2D, but under specific laboratory conditions, they can be made to grow on a third dimension and actually have volume and behave more like a muscle, like a real muscle in vitro. In this model, we have already confirmed overexpression of MBNL2 in antimiR-23B treated muscles, and also the rescue and the strong improvement in a parameter like the myotube diameter, indicating a healthier muscle and also in antimiR treated muscles. So to finish, while projecting the main funders and our collaborators, let me emphasize just four take-home messages. The first is the extensive track record that we have in the field, starting from the very beginning. Also some synergism between the research performed at the university and in ARTHEX Biotech. Also, our approach, which is new, we are attacking the root cause of the disease, but differently, not degrading DMPK transcripts. And our unique preclinical model, the 3D human muscle, that we hope that may mimic the behavior of a real muscle very accurately. And with this, Beatriz, I give you the floor.
Okay, thank you, Rubén, for this wonderful introduction. Now, as Rubén guide you through the scientific details, I'm going to give you some more details about our company, because as Rubén mentioned, we are coming from a very academic field, but we managed to jump into a company just to be able to have access to private funding that allow us to continue with the development in further stages. So ARTHEX is a spin-off of the university. We are located in the scientific park in the University of Valencia. And we have a lean startup philosophy because we are a small team, but with highly specialized personnel in different areas. Also with an important contribution of outsourced resources and advice, and with very close collaboration with key opinion leaders in the field. So well, this is the therapy development stages for every drug, something similar to what Rubén has shown you. There is always a discovery stage. And then the early stage of development involves preclinical non-regulatory studies in showing efficacy in different models, and also some regulatory studies, preclinical in different animal species, before going into the phase 1, phase 2 clinical trials. As you can see, all these stages in blue are the earliest stages of development and are the risky part, because it's very difficult that the pharmas take this risk of developments in such an early stage. So what we wanted to do with ARTHEX is to fill this gap, to move something from the university or research center into a clinical phase advanced enough to be attractive for a big pharma to continue with the development and finally with the commercialization. So what we have done is to license this asset from the university, and we are developing it until the phase 1, phase 2, where we will already have contacts with the pharmas, hopefully, if everything goes well. So this is our business model. We have our product against myotonic dystrophy. It's ATX01. We took it from the university when it was still in preclinical non-GLP development. We have working in still producing more efficacy data with the lead candidate during the 2020. And now in 2021, we are focused in the regulatory preclinical phase. And our idea is to initiate the phase 1 by the end of next year. At the same time, as we have put together a lot of knowledge about regulation of microRNAs in neuromuscular diseases, we would like to initiate new developments with the same focusing on microRNA targets to treat neuromuscular diseases. So we are just initiating some proof of concepts with other indications. This is the core management team of ARTHEX. I am the CEO and CSO. We have close collaboration with the university, but we also have our own laboratory in the university. And I have Pedro, Sheila, and Jordi helping me at the management level. We will have our Chief Medical Officer joining in July. And as you can see, we are collaborating with different other companies as advisors in IP issues, regulatory, toxicology, and so on and on. This is our board of directors and a group of board of scientific advisors. In the board of directors, we have Invivo Capital and Advent France, the two VCs on board. And in the scientific advisory board, we have some technical advisors for the part of the CMC issues. And Rubén Artero as expert in the disease itself. Ramon Enilla and Eric Markusson are expert in the chemistry of oligonucleotides, so they have been very helpful in the design of the antisense oligos. And we are also collaborating with two clinicians, which are Guillaume Bassev and Nicolas Johnson. Nicolas will talk also today. He was in fact helping us as chief medical advisor from the very beginning. So we are very grateful that he's here so he can give you more information about the clinical research network in myotonic dystrophy disease. And why we were focusing on microRNAs? Well, okay, we were working with the pathogenesis of the disease and the role of microRNAs in the pathogenesis and also the potential role as biomarkers. MicroRNAs are attractive molecules because they can target the expression, or they can modify the expression of different proteins. So just modifying one microRNA, you can modify the expression of all these groups of proteins. And this approach has advantages and disadvantages. So the main advantage is that this is not a genetic therapy. So you are not really producing a permanent modification in the genome, which could be frightening in some way. You are just modifying the expression, but it's something that occurs during a period of time that you can modulate very well. The regulation of these antimiRs is very easy. But the disadvantage is that then you will require a chronic treatment because it's not a cure. You need one advantage is that they are very highly specific. The sequence of these microRNAs and their targets are very well known, but you need to know very well which is their role in the disease to use them as a therapeutic target. In general, therapies based on oligonucleotides have predictable toxicity. So this is very useful in the development. So the risk of toxicity during the development is very reduced in comparison to small molecules. But on the other part, there are not specific guidelines for RNA drug development. There has been a lot of advances in this field in the last years, but you require a close interaction with the regulatory authorities to ensure that your development plan is adequate. And one advantage is that you can target multiple targets, but a potential disadvantage is that you will need to control the off targets of the therapy. So you need to be sure that the modifications that you are doing with this microRNA are what you really need, and you are not touching anything else. So, but in general, they are ideal to treat complex diseases. And although myotonic dystrophy is monogenic because the mutation is just in one gene, the mechanism of the disease is very complex. Although there are not yet any drug approved based on anti-microRNAs, There are already some of them in clinical trials, as you can see here. And there is none approved yet because they are these molecules are still something very new. The microRNAs were just discovered in 1993, and they first used it was as a potential diagnostic biomarker, and this was already in 2007. The first clinical trial, in fact, with a microRNA was in 2008. So they are still quite young, and hopefully there will be a lot of drugs approved based on these mechanisms quite soon. And well, about myotonic dystrophy, that's our main product. It's trying to treat myotonic dystrophy. Of course, you all know more than myself what is the disease and what is the impact of the disease in the life of the patients. So I do not need to tell you. And already Rubén made a wonderful introduction of our therapeutic approach. But just in short, we are trying, our therapy is trying to improve the levels of MBNL, to increase the levels of these proteins, just because this protein has been directly linked to some of the symptoms of the patients. For example, the myotonia is caused by the alterations in the chloride channel that is directly regulated by MBNL proteins. So this direct correlation led us to study this protein very closely. I had a video here showing the mechanism of our antimiRs, but as the audio is not working very well, I will send them to Tanya to include in the recording, but I will not show you now. I will explain you here. Rubén already give you some of the details. But just to remind you, we are targeting microRNAs here. We have two different targets, miR-23B and miR-218. And what we have observed is that in the patients, MBNL levels are downregulated by these two mechanisms, by the microRNAs and by the binding to the toxic RNA. As you know, most of the companies working on developing treatments against this disease, most of them are focused here on destroying the hairpin or avoiding the interaction with the protein, but we are focused on the microRNAs. So we are using antimiRs that are single-stranded RNA, very small size molecules that can bind to the micros and inhibit their activity. So in general, we have high levels of protein that are producing a relief of the symptoms. And this data has been already published in two different publications in peer-reviewed journals, and we are now about to publish more information. So last year we were, as Rubén mentioned, we were focused on the process of hit to lead, trying to look for the most efficacious molecule to inhibit these microRNAs. And this is some of the details of the screening we have done. We initiated the screening with more than 100 antimiRs, and we shorted out to 12 hits that we were testing in the Invivo models. And after that, finally, at the end of last year, we decided one candidate. Just something very short. In the comparison, here you can see the comparison of the previous antimiRs that we have published and our lead molecule here in orange. As you can see, the new lead, it's have a more important effect on the myotonia and also on strength. So our new molecules is at least tenfold more efficacious than the previous ones. And also they are safer. Just to show you some studies with the lead, here we were using HSA-LR. This is a well-known mouse model of the disease. Here in the mice, we were performing injections of the lead candidate at a very low dose, just 3 mg/kg. And then we studied the effect of the drug after different time points. I show you here, for example, 5, 10, 15 days. And with just one single dose, we obtained a reestablishment of strength during at least 10 days. At day 15, as you can see here in this is in red, you can follow the levels of strength of the treated mice, and it declines after 15 days. Regarding myotonia, we were reducing the levels of myotonia during the same period, about 60%. And we were increasing the levels of MBNL protein. Here you can see the quantification of the proteins in gastrocnemius and quadriceps muscles. As you can see, it's reducing after 15 days, but still highly significant in the treated mice in comparison to untreated mice, which will be here in gray. The control levels, which will be healthy mice, are represented here in black. So at 5 days, we were already normalizing the levels of the protein. And this has a correlation in splicing, because MBNL, as you know, is, as Rubén explained, is one of the main roles of this protein is splicing. So we were rescuing some of the splicing defects, such as this is the CERCA channel, chloride channel. Also, we were rescuing the splicings at different levels in the muscles of the mice. Something also important about the microRNAs is that they are very well conserved. So we have exactly the same sequence of the microRNA that you can see here in pink. In humans, mouse, rats, mini pigs, dogs, and also in non-human primates. So this allows us to check the efficacy of the same molecule through all these species. So now we have some experiments already in rats at different concentrations. We are mainly testing toxicity in these animals, but also the distribution of the oligo and the effects on MBNL protein in the muscles. So these are the effects on MBNL levels in the gastrocnemius muscles of the rats that had been treated 8 mg per kg or 30 mg per kg during 3 weeks. We obtained 5-fold more proteins in the rats treated, which was a lot. And now we are looking for, we are checking different concentrations. And also we have looked for the duration of the effect. And as you can see here with rats treated at 8 mg per kg, these are the results obtained 1 day after the injection, 5 days, 10, 15, and 21 days. And although there is a peak at 5 days, we are always obtaining very high levels of proteins, around 3-fold in comparison to saline treated rats. And this is important that we have a duration of more than 3 weeks of the molecular effects of the therapy. And the good thing about being able to follow the target engagement of our therapy is that we can adjust the doses, and that we will be able to follow also the effect of the doses looking at the MBNL levels in the biopsies of the patients. So this is the preclinical timeline. These are our homework for all this year and next year. We are now focused on the manufacturing of the oligo. We had already a first batch of 10 grams. We are now about to initiate another synthesis of 30 grams for the regulatory studies in the animals. And we have already booked the slots to initiate the synthesis of the 100 grams to be used in the clinical trial. This batch will be ready by July next year. We have also set up the conditions for the analysis of the stability of the oligo, and also for the quantification of the oligo in the plasma and the muscles of the animals. And this method will also be adjusted to the human samples when we initiate the clinical trial. And we have already done some preclinical studies in rats, and we will initiate preclinical studies also in dogs and non-human primates after this summer. And also we'll study some, we'll perform some genotox studies and phototoxicity, which are required for the IND application. We have just started the interaction with the FDA. by submitting a pre-IND meeting application. We have a meeting with them on 10th September. That will be very useful because then we will have the opportunity to validate our development plan, the preclinical and also the clinical plan that we will have by then. And we will also submit the ODD first to the FDA and then to the EMA after summer. The idea here will be that by July next year, we will have the compound ready to use in humans and all the preclinical studies needed to fill the IND. So the submission could be done in August. Hopefully by the end of next year, we will be starting the clinical trial. And although unfortunately I cannot give you more details about the clinical trial because our CMO is not yet on board, but we have already Nicolas' advice, and we have the Myotonic Dystrophy Clinical Research Network as site management organization that Nicolas will give you more details, but they will be very helpful for the site selection, startup, and patient recruitment. And they will collaborating with our CRO, clinical CRO, which is PPD, during these clinical trials. So just as an overview, our product ATX01 is a therapy with proven healing potential, at least in the proof of concept in the models of DM1, in different models. We have a new target, which is an innovative therapeutic approach. In fact, our target is easier to access than others because the microRNAs are located in the cytoplasm. We don't need to go inside the nuclei to target our microRNA. Also, our therapy is compatible with others because other therapies that are under development focusing on deleting the repeats could even have higher efficacy, combining them with our therapy addressed to increase the levels of the protein. Another potential advantage of our therapy is this multi-target approach because we have data supporting that other targets of the microRNA are involved in muscle maintenance and regeneration. So we could be doing more than increasing MBNL. We could be targeting other important pathways for muscle improvement. And we have high efficacy in the muscle tissues. We are working with our active doses are very reduced, comparable with other strategies which are focusing tissue-specific delivery, because we achieve this highly efficiency by conjugating our oligo with a fatty acid. That is we are about to submit the patent of this new conjugate. And now I give the floor to Nicolas. So I think he will give you a very important information. Let's see if I find the. Okay, Nicolas, they are yours.
All right. Thanks, Beatriz and Rubén. I'm delighted to join the folks at ARTHEX, and of course the rest of you listening to these industry updates. That it's a great idea by the MDF, and so I won't take too much time because I want to leave time for questions if there are any. Beatriz and Rubén have done an outstanding job explaining the pathogenesis of myotonic dystrophy and how their therapeutic works. This part of it really is about preparing for clinical trials and less about the drug itself. Let's just see if I can get there we go. Okay, so this is my sources of support. So, I spent a lot of time thinking about how to design therapeutic trials in myotonic dystrophy. What are some of the challenges that occur? This is a rare disorder. I think it's it's certainly at least 1 in 2,100 in terms of the prevalence, and perhaps rarer in certain areas. It is slowly progressive, and so demonstrating change over the period of time that you would expect to see in the clinical trial can be somewhat challenging. You need to measure that change consistently, so we're very dedicated and focused on getting our clinical trial sites to be reliable, consistent, and provide high quality data. And then how do you know if the drug works? What outcome measure are you going to use to measure that? So the Myotonic Dystrophy Clinical Research Network was originally founded in 2013 by Charles Stewart and Dick Moxley at the University of Rochester to develop clinical outcome measures and biomarkers for myotonic dystrophy with the support of the Myotonic Dystrophy Foundation and the Muscular Dystrophy Association. The rationale was to try and understand which outcome measures were sensitive to change, to prepare sites for therapeutic trials and prepare patients for clinical trials as well. So the study, the network has previously done a original single-site natural history study in Rochester. That was followed on by a 6-site natural history study. And then also notably, the most recent therapeutic trial in myotonic dystrophy used the DMCRN sites. And one of the things that we learned from those prior studies, for those of you who are living with myotonic dystrophy, I don't think that's a surprise if you've interacted or engaged with your family members or other members in the community. It's a heterogeneous disease. There's not 2 people with myotonic dystrophy that look the same, that act the same. That's part of the disease pathology. We understand that, but it is heterogeneous, and that provides challenges to us. The clinical outcome measures that we've chosen in the past may be done in a reliable manner, and we think things that measure walking speed, grip strength, myotonia, or respiratory function are all probably really pretty important. And one of the key measures or observations from the last 6-center study is that it seems to be that even though we talk about the change in myotonic dystrophy as being stable or linear over a long period of time, in fact, there's periods of time when the disease may progress slightly faster, and that may make those people more likely to show change during the course of a clinical trial. And the graph I show here is just a stylized graph of saying, well, the blue line would be a low measurement and it doesn't change very much, and so that's probably not a very good choice. The orange measure, that may be a good choice because you can see a lot of change, and the gray measure probably changes too quickly. So we've been very focused on looking at prior performance on an outcome measure that will predict future performance. To that end, we have a large study to characterize the severity and disease progression of myotonic dystrophy over 700 patients, and really the goal is to to look for levels of impairment using standardized measures and a patient-reported outcome, and to test for patient characteristics of baseline that predict subsequent progression. And then, of course, to complete the development of a muscle RNA alternate splicing event biomarker severity, and of course, to provide robust natural history data to support drug approval using FDA's guidance on natural history studies. So these are the sites of the current myotonic dystrophy clinical research network. So they're located around the United States and Europe, and soon to be New Zealand. We need, if you're on the call and living with myotonic dystrophy, we need your help and participation. We're actively recruiting for this study. This is a meaningful opportunity for you to participate and help us shape the course of those future clinical trials. You email Mary St. Romain or Jeannie Decterbroom to participate in the study. If you're over 18, you have myotonic dystrophy type 1, you probably qualify. And of course, you want to follow along with the MDF announcements for things like this. As Beatriz mentioned, we want to give back to the community. We want to make sure that drug development occurs successfully, quickly, and with rigorous trial design in myotonic dystrophy, so we can collaborate with protocol development, consultation, clinical evaluator training, and we can provide some of that support in collaboration with PPD, as is being done with ARTHEX and our partners there. So really looking forward to seeing this therapy and so many others move forward into clinical trials. And with that, I will stop showing the screen is what I need to do, and then to give it back to you.
Yeah.
Please.
Yeah, because it's time for questions and answers. So I will again share my screen and I will show you here which are the questions that you all sent us. Let's see. I grouped them into different fields. So we have some questions already. Can you see the screen, right? Yes. I'm sharing. Okay. Because there are already some questions that Rubén answered during its talk regarding what we were doing and what was the mechanism of action. And there was some other questions regarding to the drug activity that I would like to answer. So the first one was, will your drug stabilize or allow the body to regenerate normal functions muscle tissue? I think this one and the next one are quite linked. So the next one is, will your drug reverse symptoms of just stop the progress? So I think this is a very, very relevant question. If we will be able to improve the muscle or just to stop the decrease of the function of the muscle. So this is something that we will really have the answers when we are able to test the compound in the patients. But at least according to the studies or the results that we have in the mice, what we have observed is that the muscle was improving. So at the histological level, we observed an improvement in the muscle. So this also, we have some data telling us that in the satellite cells of the muscles, improvement of MBNL levels could allow muscle regeneration. So all this data goes in this direction, that we will be able to improve the muscle performance, but as I said, it's nothing is clear until we don't test it in patients. The next question is, are there any medications being developed that might aid with managing adult onset DM1? What about childhood onset? Until now, we have been focused on adult onset, and the first in human trial we will do will be in adults. But of course, we have also tested our compounds in models, which this DME6L model is more near the congenital part of the disease. So probably the next stages will be to go and try also in pediatric population, but this will be after we have shown efficacy in the adults. The next one will be, will your treatments last a lifetime, or will we need multiple treatments over each day, month, year? How long will the effect of the treatment last? This is also a very relevant question, because as I said, this is not a cure, so it will be, unfortunately, it will be a chronic treatment. The idea is to do it as comfortable as possible for the patients. So go for the administration routes which are less invasive, and also try to redose maybe once a month or once every 2 months. But it's something that we still don't, this will be part of the research during the clinical trial. So we now know the duration of the effect in the animals, but we will need to assess this also during the first trials in the patients. And we had two questions regarding DM2. Approximately how many years until we see an FDA-approved DM2 medication for at least some of the symptoms of the disease? Here, I just wanted to mention that in the case of the DM2, MBNL1, MBNL proteins also have a very relevant role, because they are also attached to the repeats in DM2. So the mechanism of the disease is quite similar to DM1. So we envision that the next step to test will be go for DM2 patients in our development. But I would also like to invite Nicholas to give his point of view regarding future potential medications for DM2.
Yeah, I think it's an important question, because many of the same approaches being tested now for DM1 have the potential opportunity to treat DM2 likewise our understanding of the condition is not as advanced as it is in DM1. And so I know for clinicians like myself, we're working on doing those natural history studies in DM2 so that we can prepare those patients similarly for clinical trials, hopefully in the not too distant future.
Mm-hmm. Thank you, Nick. Yeah, the next question is already answered. According to the mechanism of action of our drug, we could treat DM2 patients, but this is something that we will need to test. There are not so many DM2 models, animal models, so it's not so easy to obtain the results regarding DM2, but according to the mechanism, it seems feasible. And well, for these three questions regarding the clinical trial, I will leave them to Nicholas. What is the most significant obstacle to producing a treatment for DM1, DM2?
I mean, I think Rubén and Beatriz did a nice job outlining all of the challenges a company has to take and the path to development. One thing I'll say is I hope that, you know, if we can produce a COVID-19 vaccine in about 9 months or so, then I hope that we can see these processes. We can take some of those life lessons and move them forward. But as it stands right now, it's quite challenging and rigorous to develop that. And on the clinical side, you know, we're very interested in making sure that we design those trials effectively, which is a big issue. Understanding the natural history, understanding which outcome measures, and how to select patients. Beatriz, if you're okay, I'll just keep going on because it looks like you got the next two from me.
Yep.
How can you prepare for clinical trials right now? Well, if you're on this webinar, you're doing it already. Pay attention. Follow the MDF's news feeds and their clinical trial announcements. Please consider participating in a natural history study like NDM1 if you're eligible to do so. We have 16 centers within the Myotonic Dystrophy Clinical Research Network. Those centers and many other neuromuscular clinicians probably have a lot of existing expertise and knowledge about when the trials are going to be coming up. So those are the kind of things that you can think about. Eligibility for a new clinical trial, that's a hard thing to answer until that trial is fully approved by both the regulatory authority like the FDA and the local IRB. But typically speaking, clinical trials really do focus on those patients. These are pretty rigorous and with rigorous designs and with a focus on safety. So these are typically people who oftentimes, frankly, are able to walk and are able to, you know, keep up with the many different study visits and have the changes that we're looking for. So that's hard to answer because each trial is different.
Okay, the next one is where are the trials being done, and Europe or US? Here, as I mentioned, we are collaborating with the Clinical Trial Network. We will initiate the phase 1 in US, but our idea is also to use the sites in Europe later on, so potentially for the phase 2. You want to go with the next one, Nick?
Yeah, so typically speaking, and this is going to be a challenge for the community, I think, with so many exciting new therapies coming down the road. Can you participate in other clinical trials if you participate in a trial for the drug you're developing? The answer is almost certainly no. That typically, because we're looking at understanding whether or not that drug really treats your disease, we want to make sure that we can focus on that specific therapy. So I think you know, if you were in a clinical trial for this microRNA, then you would not be eligible to take other clinical trials during the period of that study. It wouldn't necessarily obligate you from never participating in any other clinical trial.
Mm-hmm. Okay, the next one is when will your new drug get to the trials, and also the timeline to market? As I said, we will initiate at least the phase 1 at the end of next year, and potentially we will the drug could be in the market in 2029, but this depends a lot on the results. If we had by any chance very good results, we could go through an accelerated pathway, so the timelines here could be very different. So at least we just know that we will start next year, and let's see afterwards. Will we need to be officially diagnosed with a genetic test to participate in your future trials? Yes, you will need to be diagnosed. You will need to be enrolled in the network, but we will probably include a genetic test in the patients that are enrolled in the trial, so this will not be a problem. And there were some general questions also. One, if we are optimistic about the successful drug in few years, I'm very, very optimistic. If we don't make it, any other people will do, because there is a lot of people working on the field, a lot of efforts and funding on this disease, because we know that there is really a need. So for sure, in the next few years, we will have a drug out there, and someone will make it very soon. So I'm very optimistic. And what the progress do you expect in the next 3-5 years on the therapies? As I said, for sure, probably, I'm not sure there will be if there will be a drug in the market already, but at least we will see very good results, hopefully, in the clinics. I don't know if you would like to add something, Rubén or Nicolas, here.
No, that's perfect. I mean, I think there's a lot of promise and hope right now. So I'm excited to see all the progress.
Lots of drug programs going on, so someone will have a success.
And regarding the COVID-19, of course, we started the company just 3 months before the COVID appeared in Spain. We needed to close the laboratories for at least 3 months. So we had a delay. We would have liked to start the clinical trial by the beginning of 2022. But we managed quite well. I mean, we were lucky because we were still a small company, and we could work, part of the work could be done at home. So the only problem was the laboratory was closed, but this was just for 3 months. So, well, I believe we were quite lucky yet. And the final is, what else can I do to improve my quality of life so that I can work and be productive until there is a treatment? I leave it to Nicolas. I think this is very similar to the previous question you already answered.
Yeah, just to highlight that the Myotonic Dystrophy Foundation has clinical care guidelines to help take care of myotonic dystrophy. So follow those care guidelines, see your neuromuscular neurologist, get your EKGs or heart checked, and continue to exercise and stay healthy. That's it.
Thank you. Also in the chat, I only see one question, which is why the network is not in Canada.
There was one other question just looking at that wouldn't be for me, which is what format do you think the drug will be administered to patients, a pill or IV? I don't know if you answered. Yeah.
Yes, yeah, we have most of our data until now it's intravenous, but now in the animals we are also testing subcutaneous delivery, and we have obtained some good results. So this is something that it's not yet clear. Of course, going for a subcutaneous will be much more comfortable for the patient, so that's our preferable option. But it depends on the preclinical studies.
And I can answer briefly about the network. Our network sites are tied to where our funding has been to date, and so we always have a list of sites that we're looking to open as more funding accumulates. So keep watching.
Great. Well, I don't see any more questions. Just to thank you, all of you, for keep the attention for all this long seminar, and thank you to the MDF for the organization for leaving us and be part of this, and thank you for the wonderful work you are doing to inform the patients, because I think it's really amazing to you have a great opportunity to listen to the people that are doing their work, and I think this is always very helpful for them. And yes, we are open to more questions. If you can send them to us, we will also reply in writing. So just let us know.
Exactly.
Thank you all. That was fantastic. Congratulations and really thank you on the world premiere of ARTHEX's incredible progress. It's exciting for the DM community and remarkably helpful to see the step-by-step process that you spelled out today. We really can't thank you enough. I also want to thank you, Beatrix and Rubén, for being here when I know it's after 10 o'clock at night in Spain. So we truly appreciate your dedication to the DM community, and to all of you for genuinely providing hope to the DM community and to the DM ecosystem as a whole. We definitely wish you luck in your next stages. We look forward to hearing more about the progress. And I want to just thank you again for being here today and wish you all a fabulous rest of the week. Thank you.
Thank you very much.
Enjoy your day there.
Okay.
Goodbye.
Take care.
Good night.